Anyway, thank you, Phei Er, for inviting me, as one of our speakers in this wonderful, session. Also, this is my first time experience, to have, to give a talk and virtually. There's a virtual conference. So, but very nice than I expected. Anyway, so as Pei already introduced me, also introduced the title of my talk. I want to talk about some naturally occurring bioactive compound derived nanoparticle for some biomedical application. So before going into the details of the my talk, I'm gonna please give me a sec. Excuse me. My slide doesn't go on next. Okay. All right. So as Pei already introduced, I was trained as a chemist. I received all my degree in organic chemistry at KAIST. But now I'm actually professors in Department of Biological Sciences. And if you see this slide, I received my PhD in organic chemistry, but I did my postdoc in biomaterials sectors under the guidance of the Bob Langer at MIT. So at that time, I switched my research interest from organic chemistry to some biomaterials. I learned a lot, while I was a postdoc in this lab. And then I was appointed as, associate assistant professors in Department of Life Sciences at GIST, Gwangju Institute of Science and Technology. And then I moved to the KAIST, but the similar Department of Biological Sciences. But as you know, even though I was trained as organic chemist, but most of my students, from this biological science, biology. So which means I have to work with them in some biology related some topic as well as some biomedical application. So because of that, I have been working on or collaborating with the some scientists of clinician at hospital in Korea. Also, I've been also very keen interested in translation of my technology into the clinic. So I founded the 3 startup companies so far. So actually, my career itself is kind of some integration of several multidisciplinary some research field. So now currently, I'm working on these 3 main themes: nanomedicine for imaging and therapy, particularly for cancer and inflammatory disease treatment. Or second is some peptide or protein-based biomedicine, that is nanomedicine. Also, we are working on the cancer stem cell related some field as well. But our interest or my interest, has been changed from this topic to other topic here and there. But one thing that wasn't changed, throughout my career was I've been interested in, some developing platform technology which may address a medical need, ultimately, eventually, translation into the clinic. So my main focus has been on development of some platform, okay, or the platform technologies. Let me show you, just several the platform technology we developed. The first platform we developed years ago is novel class of high affinity peptide, which we called peptide, standing for some, aptamer-like peptide. So it looks like, this kind of some bipolar shaped peptide. It has pre-organized structures. Let me I realized I didn't actually turn on my laser pointer. Sorry about that. Now you see the laser pointer here, right? Okay, so this peptide has this kind of pre-organized structures. There, at the center, there is beta hairpin structures, which is a constant region. It's consisting of only 12 amino acids. So forming this kind of very robust and stable beta hairpin structures. And then we introduced two arms at both ends, N terminal and C terminal, and construct these, two arms as library. For example, each six amino acid, so 12 amino acid can be randomized. So we are successfully, able to construct this peptide library. So by using fuzzy display based technology, you're able to, identify some protein target specific this kind of peptide molecules. So which shows, several tens of nanomolar affinity after just, first, the screening. So by using this target specific peptide, what it did was we used, this cancer, some protein specific peptide as a cancer targeting ligand, introduced them on the nanoparticle surface so that we are able to, achieve, cancer cell specific drug delivery or imaging, application. Also, we are able to fuse the cancer targeting peptide to protein therapeutics, cancer therapeutics, for example, TNF alpha. So this fusion protein was actually very effective in killing cancers. Also, this kind of peptide and antibody, the hybrid form was successfully, formed. So which showed very long-lasting some blood circulation, as well as cancer targeting and very deep penetration into the tumor. Also, peptide, this target-specific peptide per se have been used as therapeutics. For example, STAT3, peptide was used to, control the cancer. So the VEGF-specific peptide has been used to treat, age-related macular degeneration, so-called AMD disease. So anyway, so we are able to use this target-specific peptide to treat various disease. So second, platform technology we developed is polymer thin film platform. So which enable us to convert cancer cell to cancer stem cell spheroid. So once we actually successfully generated, cancer stem cell spheroid from diverse cancer cell source, cancer cell lines, as well as patient-derived primary cancer cell, then what it did was, analysis of a gene expression using RNA-seq, which give us, a variety of the bioinformatics data. Then, based on these bioinformatic data, we are able to, identify some very unique novel biomarkers that can be used for diagnostic marker or therapeutic markers. So we are actively working on this cancer stem cell field because we have the platform technology that generate, cancer stem cell spheroid from just, original cancer cells or patient-derived primary cancer cell. It's a very unique technology we have. Also, the last one is the bilirubin-based nanoparticles. But I'm gonna, explain this because this one is the main theme of this my talk today. So I'm gonna show you. So let me back to the today's main theme. So, which is integration of a naturally occurring bioactive compound into nanomedicine. So, as there are many different types of, naturally occurring bioactive compounds. Shown here is just a few of them. Just name a few. For example, curcumin, active ingredient from the curry, Indian curry. Also, resveratrol, which is active ingredient from wine. EGCG from green tea, and bilirubin, which is endogenous, antioxidant molecules. So we produce the bilirubin every day with a high amount. But anyway, the problem with this, the naturally occurring bioactive compounds is, even though, these bioactive compounds actually have demonstrated therapeutic potential in many disease models, but none of these actually successfully translated into clinic. So why is that? So we wonder, why is that? So as you know, all these bioactive compounds are poorly water soluble. Because of that, after, for example, oral administration, their bioavailability into the bloodstream is very, very low, less than 1%. Okay. Also, all these, if you look at this chemical structure, I'm organic chemist. So the chemical structure, you will easily find they have many conjugated double bond, which means they intrinsically very sensitive to some environment, which means light sensitive, which means unstable. So because of this property, it's very hard for scientists to translate them into the clinic. So how to overcome this intrinsic problem or associated some limitation? One approach may be encapsulating these bioactive compounds into some nanoparticle or liposome, right? Or you can make this some amphiphilic version of bioactive compound so that they are self-assembled into nanoparticle. Okay, so there are the pros and cons these two systems. In this case, that merely encapsulate this bioactive compound in the core of a nanoparticle. Of course, you can increase the systemic stability, water solubility. The problem with this system is you know the loading capacity of a bioactive compound per nanoparticle carrier is very low. Let's say less than 10% or something like that. Also, nanoparticle carrier per se, the carrier itself may have some potential toxicity, right? But if you look at this, the other side, this bioactive compound derived nanoparticle, which consists of only entirely consisted of this bioactive compound. For example, polyethylene glycol modified bioactive compound can self-assemble into these nano structures, right? In this case, nanoparticle per se can act as therapeutic molecules, as well as the carrier molecules for many different types of cargo, therapeutic cargo. Okay. So I'm going to show you, one typical example, what we actually overcome those some bioactive compound and the related the limitation using this pegylated bilirubin. So we developed pegylated bilirubin derived nanoparticle and then use them for treatment of cancer or inflammatory disease. So I have the conflict of interest disclosure because I founded a startup company 2018. the Felix, acting as co-founder as a board member. So because Felix is actually founded based on the this pegylated bilirubin technology. Anyway, so let me give you just a brief introduction. What is a bilirubin? So bilirubin is actually, final metabolite of a heme. As you see, the bilirubin is a final metabolite, and also the endogenous antioxidant compound is one of a bile acid. Yeah? So we produce this bilirubin, is a very large amount. So 10 milligram, approximately 10 milligram per liter of blood. So which means the bilirubin may function, biological function, important function, play very important function. But if you have too much, bilirubin in your bloodstream, you will have, jaundice signature because this bilirubin, which is very hydrophobic, is insoluble in water at all. So this actually must be excreted from liver from this, some metabolism pathway, so this enzyme. But if you have too much bilirubin under some disease condition, such as some liver disease, okay, then it cannot be excreted from liver, then become deposited in various tissue, including skin and white scleral part of eyeball. So you'll have this jaundice signature. But this jaundice signature is not a disease. It's kind of some sign you have some problem with your liver or in your body. Anyway, so there have been, some many epidemiological study on the relationship between, serum bilirubin level on and some disease condition. So anyway, to make long story short, so these people who have high bilirubin level experience a much lower risk of having inflammatory disease as well as cancer. Anyway, so what is that? Because bilirubin, it turns out bilirubin is a very potent antioxidant and also anti-inflammatory agent. So which can scavenge, various types of reactive oxygen species in our body, thereby protecting our tissue and cells from this oxidative damage. Okay? Because of that, scientists actually already, confirmed the therapeutic efficacy of bilirubin and in many different types of inflammatory disease and also metabolic disease animal model, in which, bilirubin is turned out to be very effective, potent therapeutic molecules. So now you understand, bilirubin has Jekyll and Hyde character, double-edged sword. So we wondered how we were able to, get rid of this Hyde character specifically, then so that we can, utilize that, Jekyll is good guy's property of bilirubin. So it was very simple solution because we just attached polyethylene glycol, one of these two carboxylic acid, then preparing synthesized monopegylated bilirubin, which is amphiphilic. So self-assembled into nanoparticle in aqueous medium, as you see here. So as you know, reactive oxygen species, play a crucial role in progression and pathogenesis of many different types of the some inflammatory disease, either chronic or acute inflammatory disease. So we actually tested the therapeutic efficacy of our bilirubin nanoparticle in, for example, here, some inflammatory bowel disease model. As you see here, bilirubin nanoparticle treatment actually prevent the progression and pathogenesis of IBD very effectively. Also, the in hepatic ischemia reperfusion injury model, which can be prepared by microclamping at this, vessel, so that 2/3 of the liver fell into the ischemia state. Anyway, in this model, our bilirubin nanoparticle administration through intravenous injection showed actually normalized the ALT and AST level, close to normal compared to untreated, control group, which showed a very high increase in the ALT, AST. Also, bilirubin nanoparticle were able to prevent some hepatic damage H&E staining and tunnel assay, so apoptosis is very effectively prevented. Also, necrotic area was, way high reduced upon this bilirubin nanoparticle treatment. Anyway, so in cardiac ischemia perfusion injury, which is very close to the hepatic injury model in terms of the mode of action, so which was done by Professor Peter Gang at Harvard Medical School, so clinician and cardiology clinician, according to his experiment data. So bilirubin nanoparticle treatment actually back to the normal function of the cardiac. So it is very effective, potent in preventing this cardiac IRI as well. So anyway, so due to the time limitation, I'm gonna just show you this bilirubin nanoparticle. We showed bilirubin nanoparticle could be used as universal anti-inflammatory therapeutic nanomedicine without immunosuppression. So we already showed the therapeutic efficacy in this islet xenotransplantation model, hepatic IRI, acute asthma, also the after oral administration of hyaluronic acid bilirubin conjugate, which successfully inhibit the progression of IBD after oral administration. Also, not only this disease, but also the psoriasis, multiple sclerosis, autoimmune disease model, we show the very potent therapeutic efficacy of our bilirubin nanoparticle nanomedicine. So you may wonder why this bilirubin nanoparticle showed a very potent therapeutic efficacy compared to just free bilirubin or other antioxidant agent. Here there are several reasons, but I'm gonna show you just the three key actually aspects of bilirubin nanoparticle have. First, this is an N-acetylcysteine, is a NAC. It's FDA approved antioxidant drug, approved to treat acetaminophen induced acute liver failure. It's FDA approved antioxidant. Compared to this NAC, bilirubin nanoparticle is 100 times fold highly more antioxidant power it has. Okay. Also, because the bilirubin nanoparticle is a nanoparticle, pegylated version, it can long circulate in the bloodstream when it injected. So as you see here, the half-life of bilirubin nanoparticle is almost 7 to 9 hours, which is way longer than small molecule, which mostly shows just less than 30 minutes or 15 minutes in blood circulation. This is second reason. Third reason, because of this nanoparticle character, it has ability to passively target the inflammatory site where some vascular permeability is increased, right? So bilirubin nanoparticle preferentially accumulate, of course, in a passive manner, not active manner, inflammation site. So for all these three reasons, we think the bilirubin nanoparticle showed very potent therapeutic efficacy. It's almost, so what about safety? So we confirm, bilirubin nanoparticle has any lethal toxicity, and apparent, body weight measurement or hematological or histological level. There, we didn't see any, toxicity after we injected, 60 times higher concentration than the level in our body, endogenous level of bilirubin. It's very safe. The other aspect, of a bilirubin nanoparticle has, it can be used as, some therapeutic medicine to treat inflammatory disease, but also can be used as a direct delivery carrier. But because of time limitation, I'm going to show you this bilirubin nanoparticle. Once, light irradiated, it converted to some water-soluble version of isomer is suddenly disrupted and releasing drug, the environment. Also, interestingly, this bilirubin particle is once oxidized when they encounter, reactive oxygen species, it converted to water-soluble version of biliverdin, which means this bilirubin particle once encounter ROS in our body, endogenous ROS in some disease site, it converted to water-soluble biliverdin, it also get disrupted and releasing drug outside in this manner. So to test that, we actually constructed this biotin-related bilirubin particle encapsulating doxorubicin, and so we can arrive the tumor microenvironment where the ROS is enriched. Then they react with the ROS, and the particle is disrupted, and they're releasing drug, and then released drug can diffuse into the tumor site more deeply. So because of that, we actually observed very high level of therapeutic efficacy, as you see here. Anyway, bilirubin particle also, can be constructed as a theranostic, probe, because this pegylated bilirubin, because of this, is endogenous some characteristic to able to chelate many different types of metal ion. So pegylated bilirubin can successfully coat some metal nanoparticle, surface of a metal nanoparticle, or some metal ion can be easily chelated into this bilirubin nanoparticle layer. Also, some perfluoropentane ultrasonic imaging gas can be also encapsulated in this manner. So we are successfully able to construct some therapy because of this bilirubin anti-inflammatory agent. Also the imaging because of this kind of ultrasonic contrast agent gas or copper 64 or iron oxide nanoparticle for MRI contrast agent. So we have successfully constructed able to construct this kind of some theranostic probe. Anyway, so let me summarize my talk. So bilirubin based nanomedicine. This is because of this very potent antioxidant, anti-inflammatory activity of bilirubin itself. So this bilirubin-based nanomedicine was very effective in treatment of various inflammatory disease. Also, this because of the metal chelation ability, so theranostic application was actually possible with this bilirubin nanoparticle. So novel therapeutics for hard to treat inflammatory disease was developed based on this bilirubin-based nanomedicine. And also we are able to some encapsulate the drug molecule additionally to this bilirubin nanoparticle so that they were acting as some stimuli, ROS or light responsive drug delivery carrier, as well as nano carrier itself acting as therapeutic molecules. So this is my group. The building, the backside is a newly built some library archived. Actually, Phei Er actually took a picture with me when she actually visited a KAIST. I don't know when, maybe 2 years ago, 1 year ago. Anyway, so at that time, we took a picture together here, the same, area. Anyway, thank you very much again for joining with us despite your, the very busy schedule. Thank you. Any questions? I will happy to ask, take any question from the audience.
Okay, so thank you, Professor John. Actually, today we will not have any questions from the audience, but
Oh, oh, I see.
yes, yes. But yeah, it's good to see you again. And yes, Professor John is my mentor, and he will be my mentor for life. And this is it's very good to see all the students and all the colleagues before, and I do see some old friends and new faces there. So yeah, we are very very interested in to see that how a simple idea of making something that is intrinsic to us, something that we have in the body like bilirubin, and then you have an idea and then you make it into a nanoparticle. So this is, I think, something that is for people who wants to come into nano. Nano is not some very far away or very far-fetched idea. It is actually very simple and it's very doable. So if
Yes,
you
right.
re-
Yeah.
yes, so if you have any idea, just come and talk to us. We can help you